to the third line of defense. The third line of defense is highly specific, and it is also much more sophisticated than the first line and second lines of defense. Here we're dealing with B lymphocytes and T lymphocytes. We have two types of third line of defense: antibody-mediated immunity, which is also known as humoral immunity. The antibody-mediated immunity is largely driven by B lymphocytes, and the purpose of this is to make antibodies that will target the pathogen. The next type of third line of defense is cell-mediated immunity. This is largely driven by T lymphocytes. The purpose of cell-mediated immunity is to kill cancer cells and virus-infected cells. Both of these types of immunity are highly specific, and they must actually be primed by an antigen-presenting cell before they will target a pathogen. They are not part of the general surveillance or general immunity. Before we go into the process of antibody-mediated immunity, let's take a look at the 5 major classes of antibodies. We have IgA, IgD, IgE, IgG, and IgM. The word Ig stands for immunoglobulin, which is another word for an antibody. Antibodies are proteins. IgA antibodies are found in bodily secretions such as saliva, tears, mucus, and breast milk. IgD are a special kind of antibodies that are always found attached to B lymphocytes. IgE antibodies are involved in allergy reactions. IgG is the most abundant type of immunoglobulin or antibody, and this is very important for fighting off infection when you are exposed to a pathogen a second or third time. These are very small, and so the IgG can actually cross the placenta. The anti-Rh antibodies belong to this group. IgM antibodies are very large. These are the largest class of antibodies, and so they cannot cross the placenta. The blood types A and B, the antibodies against them, anti-A and anti-B antibodies, cannot cross the placenta, and they belong to the same group. Let's imagine that a virus has entered the bloodstream. This is the antigen on top of the virus, and the goal is for the B cells to make antibodies that will exactly match this antigen. However, we have millions of different kinds of B cells, each with its own antibody. So to find the correct match between the antibody and the antigen can take many days. So let's imagine that we pick the red antibody as the chosen B cell and the antibody that correctly matches the antigen. This stage of antibody-mediated immunity is called antigen recognition, and it can take several days. By then, the virus is already multiplying and infecting the body. Even though the correct B cell and its antibody has been chosen, the antibodies cannot be made until permission is received from the helper T cell. So a different kind of B cell presents the antigen to the helper T cell. It kills the pathogen first and then presents it to the helper T cell, which produces a chemical called cytokine. Cytokines have the ability to stimulate the production of antibodies in the next few steps. This stage is called antigen presentation, and the cell is called an APC, or an antigen presenting cell. And the purpose is to get permission from the helper T cell. Now that the B cell has permission, the chosen B cell then multiplies. It forms a clone of itself. Once it forms a clone of itself, it will be ready to differentiate and make antibodies. So these are the chosen B cells, the ones that have the ability to produce the correct antibody matched to the antigen. And this stage is called clonal selection. Once many new B cells are made, the chosen B cells are made and they multiply, they now differentiate into two types of cells. One kind of cell is called the plasma cell. These are the pink cells that you see here, and they have the ability to actually make the antibody. What you see in blue is the rough ER, and if you remember, the rough ER make proteins that leave the cell. Antibodies are proteins that leave the cell, which is why you see the rough ER here. The other type of cell that is the memory B cell, and these cells have the antibodies attached to them so that they will be able to recognize the antigen again when there is a future infection. So these are memory B cells that will circulate in the body. And this stage is called differentiation, and this is stage 4. The last stage in antibody-mediated immunity is the production of antibodies. As you see, this is a plasma B cell and with its rough ER, very functional, and it's producing antibodies. The Y-shaped red structures that you see here are the antibodies, and they have the ability to target the antigen so that they may be marked for destruction by other cells, by other white blood cells. Antibodies do not directly destroy the pathogen. They do it indirectly by targeting the pathogen in a variety of ways. So let's look at them one at a time. The first is neutralization. The antibodies surround the pathogen, they find the antigens on the pathogen and attach themselves to the pathogen. So here's the pathogen shown in gray. It has antigens on it, and the yellow antibodies that we see here attach themselves to the pathogen, thereby rendering them ineffective. The second way is agglutination. It has the word glue in it, which means it makes the pathogens stick to each other. The antibodies attach themselves in such a way that the pathogens stick to each other, thereby rendering them ineffective. The third way is precipitation. It's like sweeping up the antigens that may also be toxins in the blood, sweeping them up and making them more visible. So here are the antibodies attaching themselves to free antigens and making them more visible. The fourth way is to activate the complement proteins. That's one of the most important functions of antibodies, is to turn on the complement proteins. The complement proteins, once activated, can make holes in the pathogen and cause death or destruction. The neutralization, agglutination, and precipitation make the pathogen and the free antigens or toxins produced by the pathogen more visible to phagocytes. In this way, phagocytosis is enhanced. Complement proteins, once activated, can make holes in the pathogen, causing the pathogen to be destroyed. We call that cytolysis. Remember, we said that cytos means cell, lysis means destroy or break down. The first time that you are exposed to a pathogen, your immune system takes about 3 to 5 days to produce antibodies. This is because you need to go through the antigen recognition stage, the time when the antibody and the antigen must be matched correctly. This can take a long time, and during this time, the pathogen continues to multiply. It might even make you sick, and you may show some symptoms. However, during the first round, you're making memory cells. So the second time you're exposed to the same pathogen, the memory cells recognize the antigen and they go to work. They skip the antigen recognition stage and you immediately make a large number of antibodies. So let's take a look at this through these two graphs. The graph on the left shows what we call the primary immune response when you are exposed to the antigen the first time. Here in blue are the IgM antibodies, which peak around 12 days. Shown in green are the IgG antibodies that peak even later. Because these antibodies are produced so late, you're already sick and the pathogen is multiplying. We call this the primary immune response, and it does go through the antigen recognition stage. Now for the secondary immune response. When you're exposed to the pathogen again, notice how the IgG just shot up on the very first day, producing large number of antibodies. The IgM also goes up, by the IgG brings about what we call the secondary immune response, which is very swift and highly effective. So there we have it again. The IgG is shown in green and the IgM in blue. When we talk about immunity, we need to ask a couple of important questions. The first one is, how did you acquire the immunity? Was it by natural means, that is from someone else, or was it by artificial means, by getting an injection? Was it active? Is this the kind of immunity that is active, or is the kind of immunity passive? So let's address each of these. Natural means it's obvious, you got it from someone else. Artificial is also easy, you got a shot. If you got an injection, then it's artificial. The active and passive are less straightforward. If it's active, it means that you received antigens. If it's passive, it means that you received antibodies. These two have consequences. And the consequences are, with active immunity, when you receive antigens, you end up making your own antibodies. If you make your own antibodies, you're also making your own memory cells. This means you have long-term protection. With the passive immunity, you receive antibodies. If you're receiving antibodies, then you don't need to make any antibodies. If you're not making antibodies, then you're not making memory cells either. This means you have no protection for future exposures to the same pathogen. So the protection you're getting with passive is temporary. You could even think of it as an emergency protection. Let's take a closer look at active immunity. Remember, with active immunity, you are receiving antigens. So we have two ways to acquire active immunity. It could be naturally acquired active immunity or artificially acquired active immunity. With naturally acquired active immunity, you were exposed to the pathogen in a very natural way. You got it from someone, or you maybe even got sick from the pathogen. It could be a flu, it could be a cold, it could be chicken pox. Any of these things you got because you were exposed to the pathogen in a natural way, from person to person. Artificially acquired active immunity is when you receive the antigens through an injection. When you get a flu shot, you're getting dead flu viruses, or you're getting the antigen of the flu virus that's been isolated. If you're getting the COVID vaccine, that would be artificially acquired active immunity because it's supposed to give you a long-term protection. Depending upon how long the memory cells last, you get long-term protection from active immunity. This could be from a few months to several years or your entire life. Let's take a closer look at passive immunity. In passive immunity, you are receiving antibodies. There are two ways to receive passive immunity. You can acquire it either naturally, where we call it naturally acquired passive immunity, or you could acquire it artificially, and we call it artificially acquired passive immunity. In the naturally acquired type, we're talking about babies receiving antibodies from their mothers during breastfeeding. The specific type of antibody that they receive is the IgA antibody. This also happens when pregnant mothers pass on their antibodies that can pass through the placenta to the fetus to protect the fetus in the short term. So this is a very temporary measure. The artificially acquired passive immunity is almost given as an emergency measure when a patient has been exposed to a very dangerous pathogen. A good example of that is COVID. When a patient is showing severe COVID symptoms, they are sometimes given antibodies to COVID, anti-COVID antibodies, and these provide protection to the patient in the short term. Again, this is an emergency measure. Another good example is when someone has been bitten by a rabid animal, such as a rabid dog, they're given an anti-rabies shot. The anti-rabies shot contains anti-rabies antibodies, which will prevent the rabies virus from multiplying and causing death. Now for the cell-mediated immunity. Here we have in green the antigen-presenting cell presenting the yellow antigens to the helper T cell that is shown in pink. The helper T cell responds by producing cytokines, which are chemicals with the ability to turn on the entire immune system. So the helper T cell is something like the boss or the coordinator of the entire immune system. It enhances non-specific defenses such as inflammation. It also gives permission to the B lymphocytes to make antibodies. So it enhances antibody-mediated immunity. The helper T cell also enhances cell-mediated immunity. Here we come across another kind of T cell called the TC cell or the cytotoxic T cell. The cytotoxic T cell, like the natural killer cells, kill virus-infected cells and cancer cells. However, these are highly specific and they need the input from the helper T cell in order to proceed. And remember that the helper T cell is the boss. Now let's take a closer look at cell-mediated immunity. We'll start with the cytotoxic T cell that is shown here in green. When it comes across the enemy cell, which is shown in red, it releases a protein called a perforin. The enemy cell could either be a virus-infected cell or a cancer cell. These perforins help to make a hole in the enemy cell. The cytotoxic T cell then uses this hole to introduce the enzyme called granzyme that is shown in blue. The granzymes induce the enemy cell to undergo apoptosis. In other words, the enemy cell commits suicide. This is very much like the natural killer cell. However, one difference is that the cytotoxic T cell must be activated by the helper T cell. It is highly specific and so it waits to be primed by the helper T cell. Let's take a look at how an allergic reaction is formed by the immune system. So here we have the pollen, which is an allergen. When the allergen enters the bloodstream, the plasma B cells release antibodies. They go through the antibody-mediated immunity reactions and they release the antibodies. The type of antibodies that are released by the plasma B cell, that is shown here in pink, are the IgE antibodies. If you remember, IgE antibodies are the class of antibodies that are involved in allergic reactions. So here in yellow are the IgE antibodies. These IgE antibodies are attracted to mast cells. If you remember, mast cells are the cousins of basophil. So here's the mast cell shown in blue and white. And these antibodies attach themselves to these mast cells. This happens when the immune system is exposed to the allergen for the first time. So this is the initial exposure to the allergen. Now let's see what happens when you are re-exposed to the same allergen at a later time. Now we're exposed to the same pollen or allergen for a second time. When the pollen enters the bloodstream, it comes in contact with the mast cell. The mast cell, which was previously exposed to the allergen, now has IgE antibodies still attached to it. These IgE antibodies immediately recognize, so there's the pollen attached to the IgE. When that happens, the mast cells respond by releasing large amounts of histamine. So there's the exocytosis taking place in the mast cell, and the green dots that you see coming out are the histamine molecules. As you remember, histamine makes capillaries leaky and causes itching, which is not very different from allergy symptoms. So histamine is released, and it's the histamine that causes the allergy symptoms. And remember, we are dealing with a second or subsequent exposure to the same allergen.